And chapter 7 is titled The Control of Microbial Growth. So here we have biofilm containing influenza virus. And this influenza virus, it has been involved in epidemics of flu, and that eventually, in many cases, have caused a lot of death. So we have to be sure that we understand microbial growth in order to prevent diseases. So this chapter is going to help you to do that. So some things that we're going to learn today is what terms are used in the process of control of microbial growth. We are also going to differentiate physical and chemical methods that are used to control microbial growth. And then we are going to identify mode of actions and names of common chemical disinfectants. And we are going to also be able to see what factors play a role in whether a disinfectant or a method is efficacious or not. And as well, we're going to see what are the specific structures within microbes that act as a target for the control of microbial growth. So for starters, let's look at some terminology. So this includes knowing what is degerming. And degerming is a general term that it is used to removal of microbes from a limited area. And then sanitization is the physical removal of microbes to lower these microbe counts to public health standards. And then that will be in utensils, eating utensils. And then we have this generic name, which is called biocide or germicide, which means killing microbes. And then we have this other term, which is biostasis or bacteriostasis, which means inhibiting but not killing microbes. So an example of, for instance, sanitization. It is using these dishwashers for getting rid of microbes from the utensils. Then more terms. We have sepsis. Sepsis is referring to microbial contamination. And then asepsis will be removal of significant contamination. You're familiar with this, you already practiced the aseptic technique in the lab. So an example of a clinical application is using an aseptic surgery to prevent the contamination of internal organs. So in this case, doctors, and of course nurses, health personnel, can remove from the skin microbes. Sterilization, it is removal of all life, and this, it is helpful as one of the goals of controlling microbial growth. For instance, when you are sterilizing medical instruments in order to perform a surgery or a medical procedure that requires not introducing microorganisms into the body, which in general, that's for the most part the case. In other procedures, which is kind of odd, you can kind of think about, for instance, when someone undergoes a heavy antibiotic therapy that can remove the normal flora, and that leaves the patient with very likelihood to have diarrhea, because sometimes you have overgrowth of certain bacteria in the intestinal tract. So in this case, you might want to introduce microbes through either the use of probiotics or sometimes even fecal transplants into the large intestine. And that's to make the person to be more immune. But when it comes to sterilization, basically instruments that goes into deeper parts of the body, like for instance the urinary bladder, like in the case of catheter for withdrawing urine, or in the case of catheter that goes into the blood circulatory system, like the veins, in this case, you want to have these instruments sterile or like a respirator, okay, or surgical uh, instruments. And then we have a type of sterilization that is used to sterilize the foods that we consume, like in the case of cans. This is called commercial sterilization. And the target in this case is a bacteria, it's an anaerobe, Clostridium botulinum. This Clostridium botulinum, with its endospores, can grow in the canned goods, and then when someone consumes it, these endospores will grow back in the body of the person, so it will become from a spore into a vegetative form, and then it will cause botulism. Botulism, it is a disease that leads to paralysis of muscles because it interferes this toxin that this bacteria produces with the transmission of acetylcholine so that the muscles can contract in the neuromuscular junction. So acetylcholine, it is prevented from being released by these toxins, and then the person will have flaccid paralysis. And well, in this case, flaccid paralysis refers to the act of having softness in the muscles. And then an example of how this can cause death, it is if someone has paralysis of their respiratory muscles. Not only the intercostals, like the muscles that we have in between the ribs, but also the muscles that we have in the in the diaphragm, or the diaphragm muscle. Now, when it comes to babies, so we have this syndrome that is called floppy baby syndrome. And this floppy baby syndrome, it is referring to the fact that babies, they cannot hold their head up. And this is because you have paralysis of the muscles that leads to maintaining your head erect, which will be like the sternocleidomastoid muscles. How does this happen? Well, a baby can be fed with a food that it is contaminated with these endospores of Clostridium botulinum, and like the most common known way for this to happen, it is actually giving the baby's formula with a sweetener, which in this case will be raw honey. So when you give raw honey in the formula, in that honey it is contaminated with the endospores, then when the baby ingests these endospores, this bacteria again is going to germinate, and then it's going to release these toxins, and then it's going to paralyze the muscles of the baby. And then this will be, of course, most in the stage, of course, when the baby is fed with these formulas, and the baby already has reached the stage where it can hold the head up, which is more or less like around 6 months or so. So then, recently, we saw, like a couple of months ago, in the news, an outbreak of botulism created by a formula that it was contaminated with these endospores. And they had to recall all these cans with formula. So then that's commercial sterilization. What is the target? Getting rid of Clostridium botulinum. And then we have disinfection, which is removing pathogens from an inert surface. So when you use a chemical to get rid of germs in your skin, and it has to be something that it can be used for that, you cannot use everything. So in that case, you call it antisepsis. But when you use a chemical that is used to remove microbes from an inner surface, like the countertop, doorknobs, a thermometer, that it is called disinfection. So then the difference is living tissue versus inert, non-living surfaces. And now we are going to be reminded of some of the principles that we saw in the previous chapter, which includes knowing what is microbial growth. So microbial growth is the growth of microbes in cell numbers, but not the size of the microbes. And then when the microbes grow in a surface that is solid, they form visible groups that we know as colonies. And these colonies typically come from a single microorganism. And then the rate of microbial growth can be high, and it can happen in a short period of time, especially if the conditions where this microbe grows are good, like if the microbes grows in an area where they have a good pH, the ideal pH, a good amount of nutrients, water, the temperature is ideal. In this case, you expect to see better microbial growth than when there are restrictions. And then why do we care about controlling microbial growth? Well, we care about it because we don't want to get sick. And we don't want our foods to get spoiled. That's why we care about the control of microbial growth. So now we're going to see from this growth curve that we saw previously on the microorganisms, we're going to focus in the cell death phase. And then we're going to see what microbial control agents can help to increase microbial death. So if you remember from that curve of microbial growth, we have 4 different phases. So here we have time, and then in here we have number of microorganisms. So we had here 0 hours, and then let's say here we have 6 hours, and then here let's say we have 10 hours. So everything started when you put bacteria in a medium, and then let's say that you have added their certain number, okay? So remember that we have a phase where the microorganism is starting getting acclimated to the media. Everything is new for them. They are starting to ingest these nutrients, and then they start preparing for microbial growth. So in this case, you don't increase the number of microbes. So you stay there in the log phase. And then you have an exponential growth and you reach a peak. And this will be, this is the lag phase, LAG, lag. And then you have exponential growth or log phase, in which you have a lot of binary fission. When it comes to this microorganism that goes through binary fission, and then you reach a plateau in which the number of cells that grow is the same as the number of the ones that die, and then you have a death phase. So this will be the stationary phase, and then this will be death. So then the same rate as which you have the growth rate, you have the death rate. So here we see in this graph the number of microorganisms in the right side, the logarithmic number on the left in this growth curve, and then here we have the number of hours. So you see in this red line the logarithmic scale of death. And then in here in the blue line, this is the arithmetic death in this case. And here will be actually number of minutes in which you are trying to control this microbial growth. So let's say that you have 1 million microbes contaminating an instrument, and then you put this instrument in an autoclave, which is a device that helps to get rid of the microbes. So in this case, during the first minute, you have a huge drop of the death of the microorganisms, which is 90%. In this case, from 1 million, you end up with 100 million microorganisms. And then from 100 million, 100,000, so from 1 million, you go into 100,000, so that's 90% loss of microbes in 1 minute. And then in the next minute, what you will have is actually the reduction of 90%. Of course, that will seem as very little death because of this graph, but that's when you use this logarithmic scale to point that you have a constant death, no matter if it's 90% or less. So in this case, you have basically a reduction from 90,000, from 100,000 to 10,000. And then from minutes 2 to 3, you have a reduction from 10,000 to 1,000. So every minute that it passes by, you will have reduction by 90%. Now, if you put it in a table, not in a graph, you put it in a table, this is what you will see there. You can put here, in this column on the left, the minutes that you put your instrument to sterilize, the number of minutes. And then in here, in this middle column, you can put the deaths per minute that occurred. And then in here, on the right side, you put the number of survivors. So all that you do is that every time that you're writing down here your table, what you will put is how many deaths happens per minute, and then subtract it from the initial number that you started with at every minute. So at time 0, you had 1 million microbes. And then on time 1 minute, you have reduced your population basically 90%. So you will have from 1 million lost 90,000, 900,000, then you end up with 100,000. If you sterilize this instrument by 1 more minute, from 100,000, you will reduce the population 90%, sorry. So that case will be leaving to 10,000. And then next minute, you reduce your population from 10,000 to 1,000. And then you keep on going. And by the end of minute 6, you have 1 microbe. So, based on this, what will be the course of action? Let's say that you're the technician working in a hospital, operating this autoclave, you're sterilizing the instruments for the next surgery. Would you stop the sterilization process at minute 6 and then give the instrument and then write down this is sterile? I saw you doing this, and that is true because remember what we saw before, that as long as you have one microbe left, that microbe will undergo binary fission and then give you two microbes and then undergo binary fission again and then give you four and then eight. So every time it duplicates. And then we saw before how more or less within 7 hours, you end up with 1 million microbes. Depending on the generation time, how many minutes will it take for the microbes to duplicate? So in this case, let's say that it's a microbe that replicates every 20 minutes, which is kind of the average. So yes, it takes almost 7 hours to create a little bit over 1 million. So, I mean, no matter if it's one microbe, as long as it goes, that you cannot say, in this case, you cannot say, well, I mean, the autoclave got broken at minute 6, and I stopped the sterilization process, and then you report in your sheet for sterilization, you report, well, this instrument is almost sterile. You can use it. Imagine when this microbe, let's say that they use this instrument right away, and then it was a surgery from the brain, right? When this microbe goes into the brain tissue, it finds the conditions that are perfect. Why? Because you have a bunch of sugar feeding the neurons, you have a lot of moisture, and then you have the right temperature. So the microbes will say, yay, it's my paradise, and then they start growing like crazy in the body. So, of course, we are understanding that, then that's why whenever we sterilize instruments, we go beyond what conventionally it is known for knowing that we have microbes. So then we will see later what are the actual conditions for sterilizing in different methods. Okay, so efficacy of antimicrobial depends on how many microbes you had for beginning. And then the higher the number of microbes, the greater the time will it take for elimination. So now let's go back to here. So let's say that instead of 1 million microbes, you had there 100 million. So then you have to multiply everything here for 100. So what it means, like let's say that at minute 6 you had 100 microbes, you have to go at least 3 more minutes, at least, to sterilize this instrument. So instead of 6, then you will have to have at least 9 minutes, but what the heck, a minute can actually save a life. So then you sterilize 10 for 10 minutes, so you go 1 more minute. And then again, there are protocols that tells you specifically what are the conditions and what are the times for sterilization. So, but that's an example. And then, of course, something that affects the efficacy of control of microbial growth is the presence of organic matter. So, if you have a dishwasher at home, and then you finish dinner, and then you have leftovers in your plate, like mashed potatoes there, a piece of vegetable, well, you have to pre-clean it. There is no such thing as putting that completely into the dishwasher. First, it's going to make a mess inside. And then the detergent that it's supposed to go into the surface of your plate is not going to be able to make it into the surface of your plate because there is a physical obstruction. Yes, I understand. Sometimes we are tired. It's late at night, and then you had dinner, and then you come from work, and it's a lot of things that you did. And then you say, well, I have the dishwasher, but I feel like putting it tomorrow. I'm going to just remove all the excess of food that we had there, and I'm going to grind it. And you do that, and then you leave the plates there in the sink. And then the next day, you just put your plates back or put it inside the dishwasher, and then you run it. Well, there's a problem there. Yes, you remove the organic matter, but that plate wasn't sanitized right away. And what is that is going to do is going to make these microbes that were in the plate to grow and to make a biofilm. And a biofilm, if you remember, it's a group of cells that grows on top of each other. It's a formation of layers of microbes that you will have protection of the bottom layer. In that case, the sanitization is not going to be proper because more than likely, the bottom layer is going to be adhered into this plate. So that's an example. Temperature. Well, actually, when it comes to dishwashers, they have a cycle where they use hot water to get rid of the microbes. And then if the temperature is not reached, actually most of these modern dishwashers, they run longer. And they keep running and running and running. And then you say, well, typically my dishwasher takes like between 30 to 40 minutes to do the load. But what is going on? Well, it requires that high temperature water. If it doesn't have it, then it's not going to be properly sanitized. So then you will have to take into account also temperature. And then time of exposure. Well, certain disinfectants require a specific amount of time to get rid of the microbes. And then especially if you have some dormant bacteria like endospores. So in this case, you require more aggressiveness in the terms of the disinfectant and the time. So let me give you an example. Together with the amount of microbes. Unless you are unique, and I don't doubt if you were like that, but most of us, we went through the period of puberty in which we were asked to do started to be asked to do our own laundry as part of the growing process. Well, you have to become mature. I cannot be doing your laundry all the time. And then you were like, okay, Mom, I will do it, or okay, Dad, I will do it. Well, of course, during that time, you have a bunch of homework, either in high school or middle school, and then you end up with a bunch of clothing to wash during the weekends. And then they told you, wow, your laundry basket is all piled up. And then you were like, do your laundry right away. And then you started doing the laundry. You were tempted, if you had a washing machine, to superload your washing machine. And not only you did that, but you run it in the shortest cycle. And then, not only that, but you forgot and you didn't put it in the dryer. So then that you started that on Saturday morning, and then by Monday morning, your parent or your parents discovered what you did, and they opened the door for the washing machine. Is the smell is going to be nice? No. So that's improper. Okay? So, but then everything again plays a role. Time of exposure, following the instructions. Most of the times, uh, the disinfectants tells you how much time you're supposed to leave it in there acting. And well, of course, I know I understand that sometimes I use like very tiny font, and then it's hard for you to read. Well, take a picture of that and then zoom it out and see if you're doing properly. So then here we have a graph where you have two different populations load for an instrument or a set of instruments, and you put them in the autoclave. And then again, I kind of told you already this in the previous table, but let's see in actual a graph, how does that look like? So let's say that you have some instruments that they have low population load, like in this case it's a 3-log, and then you had another set of instruments that has twice the contamination. So this is a 6-log of number of surviving cells. So in this case, it's perfect where you can see that it takes you like around 3 minutes for this low population load, and then to eliminate the microbes, and then in this case, for this other one where you have the high population load, 6 minutes. So what you can take from here is that it will take longer to kill all the members of a larger population than a smaller one, whether you use a chemical or a physical treatment, which in the case of the autoclaves is a physical treatment. Can you use both? Yes, in some cases. Why not? Like in the case of when we are sanitizing instruments, eating utensils, sorry. Well, you use a physical method, you eliminate organic matter, and even you rinse it. We kind of understand that by seeing others who do it properly. So with a spoon, a fork, a knife, you just eliminate the excess of food, and then you rinse it, and then you put it in the dishwasher, which is going to use a detergent, which is chemical treatment. Okay, so you can use combination of both. And now we're going to see what are the actions of microbial control agents on the cellular structures. So we saw in the cell chapter how microbes have a plasma membrane, they have ribosomes, they have mitochondria, they have a nucleoid where they have the DNA, and they have a cell wall. So all of these can be targets for controlling microbial growth. All you need to do is just think about what agent would be used for destroying each of these molecules to get rid of this. So, first agent will be an agent that controls the membrane permeability. No matter if it's an eukaryotic microbe or if it's a prokaryotic microbe, if you make holes in the plasma membrane, or if you dissolve the plasma membrane, you will have damage to these microorganisms. The reason for this is because the plasma membrane acts as a barrier not only to select what comes in and out of the cells, but also to keep what is important in the cells. It's like if they start making holes in the walls of your home. Or if you have a jacket during winter weather and then it's having a lot of holes. I mean, it's not like a single hole, or your pants where it's fashion to have a hole. Like if it has a bunch of holes, well, it loses its purpose of keeping you warm. Similarly, the plasma membrane keeps important contents inside the cells, and if you lose it, then in the case of cells, the cells will die. And then you can damage proteins, either channels within the plasma membrane or enzymes. Enzymes, if you remember, are catalyzers, so speed up a chemical reaction that can help, for instance, for the cells to utilize a nutrient or to keep them alive as part of the metabolism. So in this case, you can damage also the proteins, and then you can damage the nucleic acids. So, who remembers what the nucleic acids are used for? What is the purpose of having DNA, RNA? Yes? You would have a genetic code that gives instructions for the cells to make a protein, to make plasma membrane, to make an organelle, to grow, to repair. So without these nucleic acids, then we will lose these essential functions. So basically, damaging DNA in the microbial cells would prevent them from replicating. So that's another way to control microbial growth. And then you can target multiple things. Like if you use a chemical disinfectant, sometimes you can damage the lipid, and at the same time you can damage, if they have a cell wall, you can damage the cell wall, but you can damage the plasma membrane, and you can damage the ribosomes or the enzymes with these proteins, which are made out of proteins. Now, let's see specific physical methods of control, like moist heat, dry heat, and filtration, low temperature, desiccation, and radiation, and pressure. So heat. So heat, we have increase, of course, of temperature, that it is higher than the actual microbe normal temperature that it uses to grow. And this method is the most commonly used method to sterilize medical instruments, to sterilize instruments from the lab, and glassware, which are lab instruments, basically. So, mode of action. What does heat does to the cells? Well, it denatures the proteins. So some proteins have a particular three-dimensional shape. Like let's say this. This is a globular protein. And this globular protein is made out of these chains of amino acids that are folded and compacted, in this case, as a sphere. So when you denature a globular protein, you may render up like this. So it is unfolded. And that makes them to be inactive. I mean, it's not functioning. Hmm. What can be a good example of denaturing in real life? Let's see. I guess it's just like if you have like a rag, right? And you're trying to clean a spill. Of course, you can do this if it's a minor spill, like a very tiny spill. I mean, of course, you can do this all completely. You will absorb it. But let's say that you don't want to kind of be so messy. So in this case, you fold it. This will be like if you have few drops in there, you just do this, you dab it, and then you're done. This is much better, more effective than just doing this and then dry. It's more cleaner. I'm trying to give you an example. I was going to give you like an example of, well, you have a piece of clothing made of different parts, like the sleeves, the core, and then you start chopping it off, but that's not denaturing. I mean, but you understand that you need a three-dimensional shape. In this case, will be a globular protein versus something that is unfolded. This is denaturation. It happens quite frequently, not only with disinfectants, but cellular processes. And then one way then to denature or kill microorganisms is by denaturing the enzymes. And then we have a way to measure the efficacy of heat treatment. So for sterilization, we have something that is called TDP, something that is called TDT, and something that is called DRT. Thermal dead point refers to the minimal temperature that is required to kill all microorganisms in a culture in 10 minutes. So in this case, you have a set amount of time, 10 minutes. And thermal dead point, it tells you what is the lowest temperature that you can use. So that can be used, for instance, to perhaps sterilize something that requires, or to treat something that requires to preserve the flavor of foods, like pasteurization. What is the minimal temperature that you can use to destroy? Not so much applicable, never mind about that. Because, yes, in the case of pasteurization, you just reduce the number, but you don't sterilize. Anyway, so now, you have 10 minutes to kill all the microbes with a minimal temperature. That's what it means. And then we have thermal dead time, or TDT, in which you have a minimal length of time during which all the cells in a culture are killed. So in this case, it will be the time. So we can play around either with the minimal temperature or the maximum temperature or intermediate temperature. But you're trying to destroy all the cells in a particular amount of time. It can be 1 minute, 3 minutes, 6 minutes, so you determine that. So you can say, well, at 40 degrees Celsius, I kill microorganisms in 15 minutes. What is your thermal death time? 15 minutes at that particular temperature. If you want to reduce it, and you have still the same lot of microbes, so let's say that you started with 1 million microbes, and you can say, well, in 15 minutes, I kill 1 million microbes at 40 degrees Celsius. So that's your thermal death time. But then let's say that you increase your temperature. Now let's say that you go to 50 degrees Celsius. And then with higher temperature, you expect at some point that if these microbes are susceptible to that temperature, that the microbes will destroy it much quicker. So let's say at 50 degrees Celsius, you will kill 1 million microbes in 6 minutes. Well, 6 minutes will be your thermal death time. So thermal death time can vary. And then if you have measured the number of microbes per minute as you are subjecting, as you're putting these microbes in this process of sterilization, if you calculate at a particular time where you kill 90% of the microbes at a given temperature, that will be your decimal reduction time, or DRT. And DRT is typically measured per minute. So it's when you measure and you have 90% of reduction of the bacterial population. But it can be less or more than that as long as it's 90%. So in this case, we have dead per minute, and it's always 90%. So that's your DRT. So we know that based on this DRT, since every time you're losing 90%, and then here in this right column, you have the number of microbes that survive. So again, you know that by minute 6, which is this row, you still have one microbe. And in this case, you run it for 7 minutes, the sterilization process. So when it comes to heat, we have 2 types of heat processes that we use. So we have moist heat, and then we have dry heat. So with dry heat, what it means is that you don't use water. And then with moist heat, you use water. So when you use moist heat, something that happens is that you are putting water to boil, and then that water can interact with the components of the microbes and is going to start breaking hydrogen bonds that holds many proteins together in their three-dimensional shape. So another way to call denaturation, because you denaturate the proteins with this moist heat, it is known as coagulation. So coagulation is the same as denaturation. So the proteins that have these three-dimensional shapes get unfolded and they coagulate. An example of coagulation is when you are cooking an egg, like you put in a pan, you see the cytoplasm of the egg, how it is clear in color and then become whitish. You are denaturing with heat these proteins. There's the teacher. And then boiling. Well, boiling is bringing water at 100 degrees Celsius. That's the boiling temperature of water. And boiling can kill vegetative forms of pathogens, fungi, and almost all viruses. And the minimum recommended amount of time to boil water, in this case for drinking water when you need it, it is 10 minutes. That's when you kill most of the endospores. Okay? And then in the labs and in hospitals or other healthcare facilities where they have these devices, which are like chambers, you've seen the autoclave that we have in the lab. This is called autoclaving. So you use an autoclave to sterilize instruments. In this case, you have a chamber that is going to be hermetically sealed after you start the process, and then you will have a source of water that is going to be boiling internally, and then when you seal this chamber, the door of this chamber, this heat of the steam that is produced is going to get trapped. And then when you have an exposed instrument or material, like media in this case for microlab, this heat penetrates and it will destroy all forms of life. So now we have to have a specific temperature that goes above the boiling point of water. So in this case will be 121 degrees Celsius, and the pressure that it is created and by the steam of water, it has to be controlled. So there are settings in these devices that prevent you to go beyond that pressure. So the pressure will be 15 pounds per square inch, or PSIs. And in the case of most instruments, you sterilize them for 15 degrees Celsius, for 50 minutes. And then, so then autoclaves are used to sterilize culture media, instruments, dressings, medical, and lab equipment. So then there are settings when it comes to sterilization as well, because this pressure has to be released, so the steam has to go through series of pipes to release the pressure. But sometimes you want to release this pressure out of the autoclave fast and sometimes slow. You want to let it go slow when you are using like media, because in this case can be evaporating really quick. So in this case you use a slow exhaust for liquid. But when it comes to like metal, something that if you put it a lot in moisture, it can oxidize. In this case, you use a fast exhaust phase for getting rid of this. So here is a longitudinal inner view of these autoclaves. So these will be the walls of the autoclave. This will be the door. And then, of course, when the door is open, you can put your instruments. And it has this perforated shelf, and then it has a source of water that basically brings the water into the chamber, and then you will create with increased temperature. You have a source of heat like gas line, and then you have elements that makes this water to boil, and then it will go over 100 degrees Celsius, so it will go up to 121 degrees Celsius. So then you create your steam, and your steam initially is going to recirculate to actually control the pressure in here. You don't want to go beyond 15 psi's, because otherwise you have problems. These machines are actually kind of dangerous. They can explode. So anyway, through this circulation, part of it will go out to prevent overaccumulation of pressure, so you have pressure gauge. And then you have safety valves just in case. But at the end of the cycle, let's say 15 minutes, well, the pressure needs to go out. Then in this case, you open these screens or this part of the piping, so you will have an exhaust waistline that will make your pressure to go out, the steam to go out, and then your pressure starts dropping, and then you can open this chamber only when the pressure is zero and the temperature is safe enough. So not because the pressure is zero, if the temperature still is high, so you have also a gauge to tell you the temperature, that means that you can open this device and then you can get your instruments. No, part of the steam can go into your face, and that will basically create burns. So anyone who operates these machines have to be properly trained. And then how do you know if you have sterilization or not? Well, you have, in some cases, the use of sterilization indicators, which can be some strips. Like in this case, this strip has two lines, yellow lines. It tells you this word in this second line, which is not, and then here sterilized. So when something was sterilized, when it goes into this autoclave and the steam goes through these media or these instruments, and the steam actually went into these instruments, these yellow lines will change to a dark line, and this word disappears, meaning that the instrument was sterile. Why is the difference between both of them if both of them were introduced into the autoclave? Well, because this one, they wrap it around in this foil, so the steam didn't went through, and then you will didn't have a sterilization of this strip. Any questions so far? Okay. So for pasteurization, is a method, physical method of control of microbial growth by putting items that people is going to consume, like maybe juice, milk, into a high temperature at a short period of time, and then drop this temperature suddenly. Pasteurization is not the same as sterilization. It's going to reduce the number of microbes that can spoil foods. And the purpose of having this nice method, which has been important to save the amount of food that gets spoiled, it is actually to preserve the the flavor and the nutrients of some foods. So then, as you put your items into sterilization, into pasteurization, you eliminate pathogens, but not entirely the thermoduric microorganisms. Thermoduric microorganisms are organisms that are able to survive but do not cause disease. So we have two types of pasteurization. We have HTST, which stands for high temperature short time, or we have UHT, which is ultra high temperature. So in HTST, you put your items in these chambers for pasteurization, and then you have the temperature rise up to 72 degrees Celsius for 15 seconds, and then you drop it really quick. So the fridge milk that we get from the grocery stores, they have been processed with HTST. Now, UHT is basically almost twice the temperature of HTST. This is 140 degrees Celsius. The other one was 72. If it was perfectly, the half would be 70. But it's almost twice the temperature, so that's why it's called ultra high temperature. But then because this temperature is very high, and if you leave it there for a long period of time, in this case, you have reduction of the time for less than 1 second, and then you drop it really quick. So your decimal reduction time in this case is reduced for 14 seconds. And your thermal dead point has increased almost twice here. The advantage of here is for UHT is that preserves fluids without refrigeration. I'm pretty sure that you have seen cartons of milk outside of the fridge in these grocery stores, where you have them there for a long time, and then you can buy them, and then once you open them, for sure they tell you you have to consume them within a certain period of time. And that is because if you don't use them within a certain period of time, then the microbes that got introduced then will spoil your milk. Okay? So that is all for today. Do you have any questions?